Hello, this is Siavash, and I'm happy to share progress of my project that I started 6 months ago with Canadian Open Neuroscience Platform. Autism spectrum disorder, or ASD, is a collection of neurodevelopmental disorders characterized by impaired social interaction and communication and repetitive behaviors, and approximately 1 in 68 children are affected. ASD is strongly associated with genetic factors. However, it is still unclear that disease caused by single associated gene or multi-gene interaction, which is referred to as missing heritability. Structural variations, or SVs, are type of genomic mutations, and they have different kinds, but suffice to say they are the most complex regions of genomic, and we are hopeful that they can explain some of the mentioned missing heritability. Current known ASD risk genes are all protein coding genes, and it is estimated that they account for less than 30% of cases. However, the vast majority of identified mutation reside in non-coding region, and little known about their contribution, either transcriptional or post-transcriptional, to ASD. So to achieve the goal of understanding all genetic factors involved in ASD, we need to scan the entire genome using whole genome sequencing on thousands of samples. So we use a missing database for this reason, and we use that database. Accordingly, I developed a computational algorithm to detect the contribution of non-coding mutation to ASD using a deep learning approaches. So for our analysis, I divide the analysis into 3 different sections because they have different features: long non-coding RNAs and microRNAs, and even regulatory DNA sequences. As the genetic, if we look at the genetic associations, so they are kind of inherited together. So even if we look at one genetic variation that is believed to drive the association, for example, it disrupting the binding of regulatory factors. So there are many of them that are aligned in the right together. So the problem is we don't know which of the genetic variation actually is meaningful. So we need to develop a computational algorithm to predict which of the genetic variation actually is meaningful. So the best way to do that is to train a deep learning model and try to predict the impact of each genetic variation on the disease. So we input different features into our model, like including conservation, transcription factor bindings, histone marks, chromatin states, DNA accessibility, which came from different experiments, and they are available in public database. And on top of that, for long non-coding RNAs, we use the RNA binding protein binding sites. And for microRNA, we use mRNA target sites on top of that. So we use some convolutional layers and pooling, and once it's necessary, we use batch normalization and dropout layers. And at the end, we use a fully connected layer which shared across all the feature. And then using this network and using the ASD database that we have for genetic variation, structural variation, for different probands and unaffected siblings, which we use as control, so we can predict the impact that each structural variation has on disease. So using this method, so as an example, I just show one example, one long non-coding RNA we find that is believed to be important for the disease. That, as you see, like there are some features, some peaks in histone marks that they are likely to regulate the disease. And at the end, for the contribution of the project in open science, I use missing whole genome sequencing and clinical database, which is the largest genomic database devoted to autism, and it's open source. And also at the end, I will make available my code, software, and any interesting results to public through this missing database. Thank you, and thank you for your attention.